So yeah, my name is Vanessa Corby Harris. I am from the Tucson Honeybee Research Facility here in Arizona. And I'm grateful to be here with you today. And I'm also happy that I can do this virtually. I think that, you know, COVID has made things hard in a lot of ways, but this is definitely a bright spot that we're able to reach you guys from far away and meet you all. So I was asked to talk about honeybee nutrition and microbes. These two topics are kind of broad. There's a lot of movement in these areas, but you know, this is something that the Tucson lab is actually quite interested in, or two topics that we're very interested in. And so I'm going to give you some examples of what we've done, mostly using examples from my own research group, and hopefully provide some context for why we're interested in these sorts of topics. So perhaps the most important reason for why the ARS studies honeybees is that, of course, they are economically important pollinators. So we need large, healthy colonies to pollinate many of the foods that we eat. And so what we're going to do is I'm sort of breaking this talk up in two parts. First, I'm going to talk about nutrition, but then later on, I'll get into the bacteria end of things. So let's start out by talking about nutrition. So as many of you know, honeybee populations are exposed to a variety of stressors that influence their ability to grow and do their job as pollinators. In particular, we're very interested in stress from poor nutrition, although we've studied and continue to study many of these other stressors that you see here. So why do we care about bee nutrition? Well, nutrition and the lack of suitable forage is an important stressor on its own, and that's shown here at the top. But I also like it because it plays a crucial role in how these other stressors ultimately impact the individual and the hive. And so basically, nutrition affects all of these. And so if you're interested in honeybee stress, this is a good place to start with nutrition. So bees are phytophagous, which means that they do eat plant material mostly. I have a reference here though from some work showing that they are actually omnivorous. They're not eating, you know, beef or anything, but they do eat other microbes. And so, or they do eat microbes. And so I guess technically you would call them omnivores. When we think about honeybee diets, we might think about something like this, where they have access to a large and diverse floral environment. This is probably what bees evolved to utilize. And in a pristine situation, this is in fact true. However, this is not usually the case for honeybees. Usually they get something that looks like this. This looks quite bucolic in a way, but it may not be very good for bees because this floral environment contains basically only one resource really, and that's almonds. So not having access to diverse plant resources can be harmful to bees. Almond pollination, it happens early in the year, and so it can often be cool and wet when the bees arrive to pollinate. And so bees may not actually be foraging very well in this environment in inclement weather. I should also mention here that preparation for almonds usually starts in the fall and kicks up again in the late winter. And beekeepers spend a lot of time and money on fall and early spring feeding with the hopes that their hives will be nice and big and great for almond pollination. Because almond pollination happens so early in the year, these colonies often have nowhere to go after the pollination event, so they have to go to a holding yard, and that's pictured here. This holding yard can, you know, often has no flowering plants, and after coming out of the almonds, their colony population can be really, really large, and they'll quickly exhaust all of the stored resources that they have from almonds during this time. So this is not really specific to almonds, and this can really happen at any time of the year when the bees can't go from one crop to another in quick succession. So sometimes you can actually get what looks like a large and healthy colony that ends up crashing very quickly. So the need to wake up bees for almond pollination early in the year, issues with monoculture, and the potential for having large colonies coming out of a pollination contract that now have nothing to eat leads to issues of starvation. If pollen is limited, beekeepers often have to turn to artificial or commercial pollen supplements. And these supplements are really just that. They're not meant to substitute for natural pollen, but rather supplement the colony for a short period of time. And indeed, many supplements, they only support colonies for about 2 to 3 brood cycles. So what exactly are bees getting from their plant diet? Well, they obtain their protein and lipids from pollen, in addition to some trace elements from there. And largely their carbohydrate needs are met by nectar and honey. You might have heard some of the talk about these plant secondary chemicals, such as picoumeric acid. Those are also found in honey. So what do these nutrients do for bees? Well, protein and amino acids, they're used in things like growth, physiology, energy, things that require nitrogen. They're a nitrogen source. Fats are used for, their hormone precursors. They're an important part of cell membranes and of course for energy. Sugars are largely just used for energy. Phenolics, such as these picoumeric acid examples, can be used for things like defense versus different types of pesticides and chemicals. And then we also find in both of the pollen and the nectar different types of trace elements that really function to help physiological reactions proceed. So there's several consequences of poor nutrition that I have shown here. Poor nutrition, it has a negative impact on growth, reproduction, and behavior. For example, we have an increased frequency of begging. So it looks like the larvae will actually come up to the top of the cell and maybe in the hopes of attracting nurse bees. There's the accelerated development, or some of you might know of the term precocious foraging. That's when the bees sort of go from nurse to forager quickly or more quickly than they normally would. And this is associated with hive failure. Hives where the bees are starved, they make fewer nursing visits. So the nurses that are in there make fewer visits. And we have a lot of data sort of supporting many, many years of data talking about how hypopharyngeal gland size is reduced in the nurse bees from starved hives. And of course, these hypopharyngeal glands, they produce brood food that supports developing larvae and also the queen. And they're pictured here. So these are some nurse bees on top, sort of feeding larvae. There's some attendants to the queen feeding her. This is just on the bottom left is a bit of a view of where we might find those hypopharyngeal glands in nurse bees. In foragers, they degrade, so they're not as big. And then on the bottom right, we have the glands of bees that have been fed pollen or no pollen. And clearly there's a difference here. Pollen-fed bees tend to have very large glands, whereas pollen-starved bees, let's just say they go from being nice plump grapes to prunes, or I guess raisins would be they go from grapes to raisins. So the ultimate goal of the study of nutrition in our lab is basically to look at sort of this broad topic of honeybee nutrition, but always with the goal of making better choices of what to feed our bees. And so we apply this information in 2 main ways. The first is we are looking to improve existing commercial supplements, and then also to make better choices about what to plant in landscapes to maximize bee health. So some of the current projects that we're doing, we're conducting, seems like we're always conducting some sort of a field study to evaluate different types of commercial supplements. We're looking at how diet, specifically pollen, impacts stress response, sort of this concept of seasonal pollens and whether they have different effects on the bees. Lipids is something more recent that we've gotten into. And how does nutrition and climate influence things like overwintering ability and the health of hives coming out of cold storage? So cold storage is being used increasingly in these large commercial operations to help bees to overwinter, but also as a Varroa control measure. So I want to focus on this topic of lipids. And this is something I'm going to talk about for the next several slides. So where do we find lipids in honeybees? Lipids are, let's see. Oh, sorry. Yeah. Lipids play important roles in insects. Of course, energy, hormone precursors, I talked about that. Cell membranes, they're components of cell signaling pathways, and they have to do with this diapause and overwintering process, both as an energy source, but also a cryoprotectant. So where do we find lipids in honeybees? Well, this is a little of what our lab has done related to where lipids are located in the bee and how lipid stores change with pollen deprivation, these pictures here. So one thing that we find is that the head and the fat body are two major reserves of lipids in the bee. So on the left-hand side, we have a graph here of, let's see, two different types of tissues. The first one on the left is just the, oh, shoot, I'm doing that, the head capsule, and then on the right, it's the hypopharyngeal glands. And do we find a decent amount of lipid in the head total? But if you actually put this per milligram of dry tissue weight, it turns out that those glands are actually really lipid rich. And maybe that's not surprising given the difference in gland size between pollen-fed and pollen-starved bees. Let's see, you also find a decent amount of lipid in the brain. And as I alluded to, the abdomen. This is just a picture that I lifted from somebody else looking at pollen-fed and pollen-starved bees. You can see that when bees are nice and healthy and fat, they have this very milky looking fat body lining their abdominal carcass. Let's see here, I can't see. And this is just a graph showing the difference in abdominal lipids in bees that are fed or not fed pollen. Okay. So that was the fat in the bees themselves, in the actual bee tissues. But what about the fats that are found in pollen? Well, it turns out that both the content and the type of lipids can vary across plant type and with season. So this is a plot from a publication by Anthony Vaudo working with Christina Grosinger. And we have different types of pollens here. These are both bee collected pollens like corbicular pollen loads or single source pollens from gathered from certain plants. And we've just plotted out the micrograms of lipids that they contain per unit of weight. And you can see here that total lipids vary a lot across pollen types. We also know that particular types of lipids like fatty acids can vary across plant types. So again, this is particular types of lipids, not total lipids. So here I'm showing you the fatty acid differences among plant species. They're just listed on the left there. And we're just looking at sort of differences in the number of fatty acids that they contained. So the number of different types of fatty acids they contain, but also the quantity of specific fatty acids. Let's see here. So we see, for example, because the number of fatty acids differs, there could be some fatty acids that are present in some types of plants, but not others. And then some fatty acids like linoleic and linolenic acid, they're present in all pollens, but in different amounts. And we also, I will also direct your attention to the fact that they differ in their ratios to one another, depending on the plant taxa. And so we see similar variation. So these are single source pollens, but we see similar variation by in corbicular pollens collected by hives in different regions and with access to different floral resources. So for example, these fatty acids vary with season. This was some work that we did looking at the importance of seasonal pollens in meeting the nutrient needs of bees from the spring or the fall. And although we didn't observe any differences in total protein or lipid levels between the spring and spring or fall pollens, we did see that certain types of lipids, certain species of lipids did differ. For example, per unit of corbicular pollen, which is what I'm showing you here, the spring pollen has more of this omega-3 fatty acid than fall pollen, and that's what the asterisk means. So the spring has more of this omega-3 fatty acid than the fall. And so after looking at the nutrients in pollen, our next step in this particular study was to ask how the bees respond to these different pollens, which is actually, really important. So we collected bees in the spring and fed them pollens that we had collected from the spring and the fall. And then again in the fall, we took fall bees and fed them those same spring or fall pollens. And so in terms of hypopharyngeal gland size, we saw that spring bees were more sensitive to seasonal pollens than fall bees. So that's why you see that little asterisk there. That's basically the average size of these little, let's call them the grapes in the bunch of the hypopharyngeal gland. So in the spring bees, these acini were larger if the bees were fed spring pollen compared to fall pollen. And then in fall bees, there was really no difference or no significant difference in acini size for bees fed spring pollen or fall pollen. So we hypothesized that spring bees needed the nutrients found in spring pollen, possibly because they're going through a period of intense brood rearing and expansion during the spring, during this time. And this, of course, because they're rearing so much brood, would require nice, big, healthy glands. And in contrast, in the fall, hives are contracting and brood rearing slowing down. So the fall bee glands may not be as sensitive or responsive to diet or diet type, let's say. So it was also interesting to see that the spring bees fed spring pollens, which were actually higher in this omega-3 fatty acid, have larger glands. And this is something that other folks have found too, that these omega-3 fatty acids, when they're prevalent in the diet, that means that bees that feed on that will have nice, big, healthy glands. Okay, so why do we care about essential fatty acids? Well, I do want to make the case for essential fatty acids because I'm going to talk about them in a little bit, but first let me define what they are. So essential fatty acids are types of fatty acids that absolutely must be obtained from the diet. In other words, it's essential that they obtain them from the diet. And really for bees, there's two essential fatty acids, the omega-3 and omega-6 fatty acids, and we find those in pollen where all lipids live. So it turns out that there's a lot of recent work showing that these guys are crucial for bee health and behavior. So for example, they have a role in learning acquisition, survival and brood rearing. And it's not just the absolute or the individual amounts of these fatty acids. So for example, diets higher in this omega-3 fatty acid are beneficial, but it's also the ratio of the two together. So diets that have a lower omega-6 to omega-3 ratio tend to be more beneficial. Okay. So one curious thing that we find is that supplements or commercial supplements are actually deficient in these essential fatty acids. So these are results from a quick survey of different commercial pollen supplements, and also a quick survey that I did of 3 different mixed pollen sources. So this was corbicular pollen. I'm also showing you the levels of these nutrients found by other researchers. This is sort of cited here. Also, I'm happy to pass along any of the papers that I've taken this information from if anybody's interested. So let's focus on the 2 fatty acid related factors that seem to influence bee health. And in particular, we're going to be looking at the omega-3 fatty acids and the ratio of 6:3. So first, here's the omega-3. You can see that compared to what we find in natural pollens, there's not that much of this, this alpha linoleic acid or linolenic acid or omega-3. Also, focusing on this ratio, those ratios are very different between what we find in supplements and natural pollens. In fact, in the next few slides, I'm going to be referring to these as being either unbalanced or balanced. So balanced would mean that they are closer to 1, and unbalanced means that they're far from 1, because around 1 is what their ratio is typically in natural pollen. And so this will help you as I start to get into some of the work done by my postdoc, Meg, who is on the call, so I'm sure she's happy to answer any questions. But what I want to tell you a little bit about is some really interesting work that she's doing looking at fatty acid balance and how this balance plays a role in important behaviors in the hive. So hygienic behavior is a trait that many folks are interested in. We select for them, and you know, if you look at different types of queens, a lot of times they talk about they have this lineage of, you know, hygienic parents or this hygienic lineage. So really what this behavior is that bees that are, that perform this hygienic behavior, they're able to locate, uncap, and remove disease brood. And there's a little picture of that shown here. This trait is selected for as a part of different breeding programs going on. You might have heard of the Minnesota hygienic lines or the VSH lines, or I think it's also the Russian lines. Let's see, they're selected for this behavior. However, these behaviors, mostly because they're complex behavioral traits, it turns out that they're influenced by a lot of different genes that act together, but they are also heavily influenced by environment, okay? And so this comes out as having something called low heritability. So there's a low ability of the selected genes to come through in the trait. So it's difficult. You might get different, the trait might come out differently depending on the situation, let's just say. Let's see here. The interesting thing about hygienic behavior, there's a lot of work done on this. One thing that's kind of interesting is that hygienic lines are no better than unselected lines at discriminating between Varroa brood odors from healthy brood odors. So this tells me that maybe we still have a lot of work to do when it comes to hygienic behavior. Let's see. So how are we tying this back to fatty acid work? Well, Meg developed this idea where that was very much motivated by the literature. So she sort of read that these omega-3 and omega-6 diets affect olfactory learning, but to floral odors. So these balanced ratios that I talked about before increase the ability of the bees to learn and discriminate these different types of odors. Whereas bees that were fed unbalanced diets, it has the opposite effect on cognition. So they can't learn these odors. Let's see here. There has been a lot of work on this topic, not necessarily really just the hygienic behavior. This was from Marla Spivak's lab, where workers were able to discriminate between chalk brood and healthy brood odors, but they were actually, they learned chalk brood odors better. So this suggests that it's an important odor cue in the hive, which makes sense because these workers are tasked with keeping the hive productive and healthy. So Meg wanted to know if balanced and unbalanced fatty acid diets affect the worker's ability to discriminate in-hive brood odors. So if this was true for floral odors, do we see it for in-hive brood odors? Are bees fed these balanced diets able to perform this behavior better? So she just completed a series of experiments using caged bees, but really her ultimate goal is to ask whether these results translate to the hive. And so we tend in our lab to start out many of our experiments in cages because it's a very controlled environment compared to the hive, but we realize that that's always important to test these questions ultimately in a hive. So Meg is actually doing experiments right now that test how diet influences hygienic behavior in the hive, but I don't have any results to show you just yet. But with that in mind, let's talk about these cage experiments. So Meg started her experiment by following protocols established by the Shafir lab in Israel, who have done a lot of this work, following their protocols for manipulating diets. And so they involve basically using a soy-based protein patty with different types of oils mixed in. These data that I'm showing you here represent the chemical analysis of the unbalanced or balanced patties that Meg made using these protocols, looking specifically at these important EFAs, omega-6 and omega-3. So corn oil has a lot of omega-6 fatty acids. And so when we mix this into patty, the patty ends up having a lot of omega-6 fatty acids and fewer of the omega-3 fatty acids. Flaxseed oil, in contrast, has a lot of omega-3 fatty acids. And so when you mix this oil into the patty, it has more omega-3 fatty acids than what we typically see in the soy patty alone or in the unbalanced patty. So in other words, the omega-6 omega-3 ratio is high in the unbalanced patty, so it's further away from 1. And the omega-6 omega-3 ratio is lower, or closer to 1 for the balanced patty. So the good thing about working in a nutrition lab is that we have a lot of pollen in our freezer from different projects. So we took a look at these and saw that some of those pollens were also balanced or unbalanced. So Meg was able to use these pollens as an additional treatment in her experiments. And the hope was that she could recapitulate with our soy-based diets what we might see for bees fed balanced or unbalanced pollen. So Meg fed the bees their diets in cages for about 5 days, and then she tested them for how well they learned damaged or healthy brood odors. And she harnessed them in straws and tested whether they exhibit the PER or proboscis extension reflex response to these different odors. Yeah, so you can see them doing it here. And then you can see You can see it happening there. They're getting the reward, the little sucrose reward. They're extending their proboscis after they've been presented with this odor. Okay. So bees were rewarded with sucrose as they extended their proboscis to the damaged brood odor, and then they were punished with salt water if they extended it for the healthy brood odor. And so this reward and punishment strategy teaches them what they should be responding to. And so if they're good learners, they will quickly learn to extend their proboscis for the damaged brood and will keep it, that proboscis in their mouth when they're presented with a healthy brood odor. And the same bee is tested again and again across multiple trials. We have multiple bees again and again when they're presented with different types of odors. So here's how they respond to these healthy and damaged brood odors. So these graphs are showing you that for each trial, bees quickly learn to extend their proboscis for the damaged brood. So that's the sort of this dashed line. And they keep it in their mouth, or they don't respond. They don't have this PER response for the healthy brood odor. And at least this is the case when they're fed the balanced oils diet. And when on the left, you see the unbalanced, and they're not performing, or they're not extending their proboscis differentially for those two different odors for the unbalanced diet. Okay, and we see a similar pattern when bees are fed balanced or unbalanced pollens, suggesting that when the omega-6 to omega-3 fatty acid balance when that diet is balanced, they're able to learn these damaged brood odors. So the conclusions for this nutrition part is that fatty acids may play an important role in workers' ability to learn and discriminate odors, and this might be linked to hygienic behavior. We're testing that out now. Fatty acid enriched, or maybe even just lipid enriched pollen patties may improve colony health. And really, what I want you to come away with from this first part of the talk is that lipids are an underappreciated facet of bee nutrition. Okay, so I do want to talk about bee bacteria. So let's just get into it. So I'm going to pivot here a little bit to some of the work that we've done related to bacteria in bees. Most of this work was actually done while I was a postdoc several years ago, but I also want to highlight some of the recent work that has come out of our lab and also other labs since those initial studies were performed. So most of you know probably that microbes are really everywhere. And this is no different for honeybees. So for example, your body has like crazy amounts of bacteria. There's bacteria in soil. And then this is just like sort of like one of those deep sea vents. And it turns out that there's even bacteria there. So they're really everywhere. The honeybee and the honeybee hive, they actually harbor a very fascinating array of microbes. And at this point, they're pretty well characterized. Note that while some of the bacteria are found inside of the bee, like for example, in their guts, others are predominantly nest bacteria, hive bacteria, or those that are found in the environment outside of the hive that do make it in every now and again. So in a few slides, I'm going to talk about one of these hive bacteria, which has changed names. So at first, I think it was alpha 2.2. Then it became P. apium, or Parasaccharobacter apium. But now it's known as Bombella apis. So it was reclassified, and that paper is now in press. So hopefully this doesn't get too confusing with the different name changes. So why should we care about the bacteria that live in bees? Well, they can be harmful or beneficial. So for that reason alone, maybe they could be useful. They're indicators of colony decline or stress. So for example, stressed hives tend to have the wrong balance of these bacteria, and that's also known as dysbiosis. So they can be an indicator of stress. Some bacteria have dual roles or variability within groups. So they can actually alternate between being beneficial or harmful depending on the location and the environment. And so it seems like in this case, context can be important. So because I'm interested in nutrition, one of the things that I've worked on in the past is whether the bacteria that we find in the hive environment can be utilized. And so are these beneficial, and can we use these potentially beneficial bacteria to improve bee diets? So another way to say this is that, can we develop probiotics for honeybees? And so probiotics, they have for a long time been a major component of both human and animal diets. So we're no strangers to them. And just moving back to this figure, I'm going to spend the next couple slides talking about this Bombella apis bacteria, which again is found in the hive, but rarely in the gut. And we find it to be quite prevalent in things like royal jelly, worker jelly, the nurse hypopharyngeal glands, and also larvae. So really we would call it a hive bacterium rather than one of those core gut bacteria. In particular, it's found in young larvae. It's very abundant in young larvae, and that's what these graphs on the left are showing you. Blue is Bombella apis, and the other colors represent other bacterial taxa that we find in different instars of bees. And that's actually really interesting because it's the young larvae that are fed exclusively jelly, and that, of course, is secreted from the hypopharyngeal glands, where we also find Bombella apis to be very prevalent. I also want to draw your attention to some of the work we did several years ago that was published, in 2014. So these experiments involved in vitro rearing of honeybee larvae from second instar larvae to pupae. And we observed that when you supplement the in vitro diet with this one C6, so these are just different strains, A29, B8, and C6. Those are different strains of this Bombella apis, again, formerly known as P. apium. So when we supplement their in vitro diet, in particular with this one C6 strain, those bees, those larvae survive to being pupae more so than the controls. And we had two controls. The one was the negative, which was the in vitro rearing diet without any bacteria. And we also had another control, which was the diet with just E. coli, which is something that you don't normally find in beehives. So in the case of this B. apis strain C6, it seemed to benefit bee survival from larvae to pupae. So again, we like to complement our lab assays with hive assays, and so that's what we did here. So we had we had about 50 packages that we installed into their hives, and we actually did this in greenhouses to control their temperature a little bit, because obviously Tucson is hot. It was in the middle of summer. We started applying treatments in June of 2014. The treatments were applied each week and left on for 3 days. So we either fed them patty mixed with B. apis or patty with nothing in it, just the patty. And the bees seemed to like it, they ate it, and that's what this picture is showing you. So importantly, we were able to show that this bacteria goes from the patty to the larvae. So this is just the number of Bombella apis colony forming units per 100 larvae sampled. And when we plate that out, we see that in the hives fed the bacteria, you get a lot of bacteria, whereas in the hives where it's not fed the bacteria, you don't. Interestingly, we found that B. apis reduces nosema burden by about 40% when the patty or when the inoculated patty is fed to hives. So we had 10 hives per treatment, whether it was bacteria fed or not. And then we took brood frames out, we emerged the brood, and then challenged them with nosema. And this is what that looked like. If they've been supplemented with this bacteria, they have fewer nosema spores in their gut at 10 days than those were fed the control diet. It turns out that it's important that it goes through this hive environment to have the sort of anti-nosema effect. So if you just feed bees, newly emerged bees with pollen patty that's either inoculated with this or the control, and then you challenge them with nosema, it turns out that that the bacteria actually increases the number of nosema spores that you see. So in this case, that was, context matters. So this is actually, I do want to mention this because indeed, we see that further work from Kirk Anderson's lab, who was my postdoc advisor. It shows that under different in vitro rearing environments, this B. apis does not afford a benefit compared to the control. That's what I'm showing you here. So why? Well, let me just kind of walk you through this a little bit. So looking closely at their paper and after talking to the lead author, their in vitro rearing method was actually quite gentle compared to the ones that we used in our earlier projects, the graphs that I showed you. And so as you can see, the good news is that in their negative control, which is all the way to the right, you have near perfect survival of the larvae. In our experiments, you had about 70% survival from larvae to pupae. However, the downside of this is that when you have nearly perfect survival, you can't really test whether bees can survive any better when you inoculate them with bacteria. In other words, bees can't survive better than perfectly. And so it makes sense that survival is not any better than the control when those bees are also fed B. apis. However, one additionally important piece of information is that larvae challenged with this bacteria, this M. plutoneus, which is the causative agent of European foul brood. If you compare ones that are fed with Bombella apis now, or not fed with that, there's no difference in survival to that particular, this is my cat, to that particular challenge. So to conclude this section, couple takeaways. Bees live with a lot of different types of bacteria. These bacteria can be harmful or beneficial. And beneficial bacteria may improve bee health in important management situations, like when we are trying to feed them certain diets, for example, as probiotics. But it turns out that the context in which this is done is actually very important. And so basically, when we're thinking about probiotics and bacteria, read the fine print. I just want to thank everyone. Hopefully I haven't talked your ear off too much. I hope I've been able to give you a little bit of a taste of what we do in my lab and at our Tucson honeybee research facility more broadly, and also give you something to sort of chew on as you're thinking about honeybee nutrition and bacteria associated with bees. And well, I guess with that, I will close. And if anybody's still with us and has survived this, I can answer questions.
Yeah, this is Phil. I was wondering a little bit about the whole concept of bacteria. Different hives smell differently. And so I'm wondering is that something that is, I don't know, is it in the hive? Is it in the colony? Does it change with colonies? Different colonies seem to have different smells.
Yeah. So that's probably something that has to do with the microbial environment. That could be bacteria, but it could also be fungus, some sort of fungal population. And basically every hive has, it sort of has the same type of bacteria, but in different abundances. So there could be different balances of bacteria in that particular hive that might be influencing that. So that would be my guess. Let's see, there's some questions. Oh, I see is that Leslie?
Yeah, hi, I have a question.
Sure.
I was, so in regards to probiotics or, and maybe treatment of fungal infections like chalk brood, there's a product that Man Lake sells called ProDFM. And I was just looking it up because it's got yeast and B vitamins, and it's talking about the need for that to, I guess, help with the fat body formation. So I was just wondering if you have used that product, or do you know anything about it, about the contents and the effect on honeybee fat and nutrition?
Right. So I should say that a lot of these things are proprietary, so they don't actually, a lot of times they don't tell me what's in them. I have talked to some folks about, what their mixture is. I guess I'm always a little bit cautious with that kind of stuff, because I feel like there's many times when they can't really back up what they're saying with actual, research. And then in some cases, they actually do refer to papers, and the papers aren't that great. So, so I guess I can't really comment on whether I think it would work. But there is some evidence that, certain types of bacteria actually are beneficial in terms of, those bees having larger fat bodies and things like that. So, so I don't know if that's the best answer to your question, but I can't really necessarily endorse that product, over any others. So.
Okay, well, then I guess a follow-on question is because I have been having trouble with chalk brood in the past. It's not so bad right now, but do you know of any research showing that there's particular probiotics that would be helpful to combat fungal infections or things like chalk brood?
Right. So as far as chalk brood goes, I think chalk brood is, in my experience, that's not really something that you're going to be able to clear up with a bacteria. I feel like chalk brood is, and many of you probably have opinions on this, so please chime in. I feel like chalk brood is kind of a symptom of, sort of an unbalanced age structure in the hive. We often see it in our packages, like if they don't take off, and the chalk brood tends to come in pretty quickly if those packages don't, increase in size, in population size. So I'm not really sure that the chalk brood would be influenced by the bacteria, but I think that it's more influenced by the age structure of the hive.
So essentially, you're saying that a age structure that's skewed to the older ages is more likely to have chalk brood.
I think so, yeah, because there's not many people, there's not as many bees in there that are performing, those cleaning tasks. Those bees tend to be the younger bees. So even if some of those older bees are trying to perform those cleaning tests, they might not do a very good job of it. So,
Thank you.
yeah. Okay. I see Eleanor, but she's muted. Let's see. Maybe she can, I saw her hand up, but.
It was the same question about strong microbials, which is a similar product to the DFM.
Yeah, I know that one. And we haven't seen any, we've done similar cage studies where we've, mixed that into the diet and looked at nosema levels, for example, and we don't see any difference. Of course, that's in a cage, but we don't see any differences in nosema levels between the ones that have been fed that versus the control. So yeah, I think when it comes to probiotics, it's still very snake oily in a way. So I would just be a little bit cautious with that. Is that wrong? Sure.
Me? Are you
Yep.
asking me? Okay. So if I added flaxseed oil to a pollen patty, really kneaded it in, would that be the good idea?
Well, maybe Meg can answer that. My postdoc, she's on here. Meg, can you unmute yourself?
Hi. So whether you can do it, you totally can. I actually knead the flaxseed oil into the patty myself, like by hand. Sometimes I use just like a small mixer. Whether it's, going to be effective at the high level, I don't know. I'm actually looking at that right now. But our cage studies suggest that it impacts learning in nurse bees, and they can't tell the difference between damaged baby odors and healthy baby odors. So that's intriguing. So maybe next year I'll have a paper for you, and that could better answer your question.
So right now I've got two very, very small recent swarms. So they're small because they're late. And so I'm feeding them pollen patty, and they are really digging it. But I thought if I add a little flaxseed oil to it, that might help them even more.
I looked at survival in the cages to see if any of these diets cause higher mortality, and I didn't find that any of these oils caused mortality in these cage experiments. So, I don't want to say, well, it can't hurt, but I can kind of say, it probably won't hurt.
Okay.
But I can say that just when I've fed the patty at the hive level, the bees seem to prefer patties with pollen in them. I have to actually spike the cooking oil patties with pollen, and they smell different. They don't even smell good to me, really. Like the pollen patties smell much better. So
Okay,
I do notice that they like to eat the patties that have pollen in them, but if you just put the cooking oil in, they, aren't as stoked on it.
Yeah, okay, right on. Thanks.
Yeah.
I wonder, does it make sense to ask the question, is it a matter of a minimum of omega-3 and a minimum of omega-6 versus a ratio? Year one?
Right. So the actual, it's both. Okay. So you do need you need to get to sort of this like critical level of the omega-3, but if you're doing that with something that's also going to increase the omega-6, that's not good. What we want is both a higher absolute level of omega-3, but we also want more omega-3 compared to, an equal amount or near equal amount of omega-3 compared to the omega-6. So that's why things like flaxseed oil that are high in omega-6 will actually achieve that without fooling around with the omega-6 levels too much.
So then what we really want but don't get is an assay of the product that we're likely to buy that says what the omega-3 or omega-6 is. And if we knew that, we could supplement it, or if someone published the assay, then we would know how to supplement it.
Right, and so I will that paper, I'm going over the reviews now. And that
Oh,
information,
good.
yeah. But what I can tell you is that they're all deficient. It doesn't matter, like they all stink, you know, in terms of omega-3. And actually some of them stink more than others. And so, you know, maybe you could make the argument like, well, I'll pick the one that's, you know, maybe a little bit more towards that 1-to-1 ratio, but really they all stink. And I
Well,
think
that's saying then add flaxseed oil to everything, but can you over add flaxseed oil?
I think you probably could if they didn't, you know, you would know because they won't, you know, because they won't find it maybe palatable. Even, you know, when the ratios are kind of more, more skewed towards 3, they still have good, they still have increased brood rearing ability, larger glands, good learning compared to if there's a diet that's heavily skewed towards the omega-6. So
then mix various concentrations with of flaxseed oil, see which one the bees prefer, and don't use the ones or mix down the ones that have too much flaxseed oil.
yep.
Got a formula that might work.
Yeah.
Is almond pollen well balanced?
Great question.
No.
Okay, then what should the almond growers be planting as a supplemental source of blossoms to improve the health of the bees?
Yeah. There are definitely some things that are better than others. Things like, let's see if I can remember off the top of my head. I'm struggling to remember exactly. Things, actually a lot of like stuff that is sort of like a canola type of thing, from the brassica family tends to be pretty high in omega-3s.
Well, the trick is something that will be blooming while the almonds are also blooming.
Right, and so brassica, especially like rapeseed and canola, they tend to be cool weather forage. But also, you know, if you find yourself in a situation where there's a monoculture that, you know, is very unbalanced and you kind of want to skew things, things that fall in like the clover family are actually quite good with regards to trees. And I'm happy to share that with you because if I'm, you know, I'm happy to share those papers with you because there's actually some pretty decent work not done by myself, but you know, these folks in Israel looking at those the single source pollen, and I'm just email me, I'm gonna send it along.
Maybe I've seen some cover crops go in there for that are basically like mustard. Mustard seems to be the only thing that really seems to bloom really because it's a February that almonds is. You know, we normally do almonds like the first or second week of February.
Yep. Yeah, the mustards are cold, they bloom and they grow and they bloom in the cooler weather. So
yeah, they're some of the first things that bloom in that area.
Yeah. Let's see.
So I got a question for you, Vanessa. So, you know, almonds is a very short crop. It's about a month long. So you know, some years it's shorter, very seldomly a little bit longer. As long as you move out of that monocrop within that month period, is there much harm to the bees? We move ours out to, you know, they're on wildflower basically after almonds.
Yeah, if you can pop them on some forage, that's great. If they're coming out and they're just going into a situation where there's nothing to eat.
Yeah, the migratory guys stage them and they'll put 400 hives in one location, and yeah. So what I was wondering, I mean, we're not that big, but we normally will come out of almonds, and the bees are huge come out. I mean, they grow quickly in almonds. and we go off onto other forage, which is for us is wildflower. We might move over to cherries. We did cherries this year, but do you see any problem because of the shortness of almonds, you know, with long-term bee health by moving them onto you know, a varietal forage?
No, I don't. I think if you're moving them onto, especially like, you know, mixed, you know, wildflower situation, then they should do okay. Nutritionally, I mean, you know, there's other problems that might be introduced from, you know, the pesticide situation or something.
Has problems.
Right. So I can only really speak to the nutritional aspect of that, not necessarily the sort of pesticides aspect, but or other things. But yeah, I mean, if it's you know, a variety, and if you're moving them into something, that's good.
And how about, has there been any studies, you know, we were in cherries, and, you know, the cherries were in full bloom, yet they were still going off onto, you know, again, there was lots of, there was lots of mustard and other flowers growing at that point in cherries, because cherries is after, you know, just slightly after almonds.
Mm-hmm.
Do you see any, is there any studies on, you know, them automatically going after a variety, even though they're sitting in the middle of a cherry orchard? My observation was they were going also to the wildflowers as well.
Right. I mean, I would say that's a good thing. You know, if they can have a very diverse, you know, source, you know, floral resource, that's good. I mean, I don't know that. I know that some folks up, Teresa Pitt-Singer at the Logan Bee Lab in Utah, she kind of did some work with Neil Williams about, you know, asking the question of if it's bad for pollination to have these, you know, other sorts of resources around. So I'm kind of extrapolating here for cherries, but I don't think that they saw any difference in pollination rates of their almonds. So if we can say that that's still the same case for cherries, it's good to have a lot of different things for them to eat, even though the number of visits, you know, might not, they might be visiting other things, but that's good. You know, they want this diverse diet.
Right. Okay, all right, thank you.
Sure.
Well,
I see, oh, sorry.
Go ahead.
I see that Sung had her hand up.
Yes.
Thank you. It's, yeah, probiotics has been on one of my back of my mind, and I have participated in some of the project, and it was a phenomenal. But I have one question about the chalkbrood. When we're speaking of that, any textbook, any protocol, when you look at it, if the chalkbrood persists, then it says requeen. And can you explain why it needed? And then what does it do? And then any queen, replacement queen will do the job, or it has to be something specific.
Right. So I think there, in some ways, there's a danger to requeening, right? But I think the whole idea behind that is that you're going to have a stronger queen that, you know, is more productive. And so eventually when you have that age structure, you know, more bees being produced and that age structure tends, you know, starts to even out to where it should be. That's the purpose of the requeening. However, when you requeen, there's a period of, you know, broodlessness, right? So there's a danger to that too. So I think they're probably, you know, looking at the cost versus benefits there. But ultimately, you know, in our experience, and, you know, this is just in Tucson, can be different other places, that chalk brood is very much a, you know, a symptom of bad, you know, age structure in the hive. So I think that's where the requeening comes in. So I've bent your ears for a long time now, so I'm happy if I need to get cut off.
Well, that's okay. I do have a couple of other questions, and I see Nikki's unmuted, and I wonder if she has a question.
No, I just unmuted because.
Okay. My question is, if there's pollen coming in and there's stored pollen, will the bees just ignore the stored pollen and only use the new pollen that's coming in?
That's a
Even
good question.
Even
That's a
if
good
the pollen
question.
is unbalanced?
Yeah. Oh, okay. So the unbalanced aspect I can't speak to, but right. So that's an interesting facet of that question.
Oh, another project.
Right. Yeah. So, okay. So I do know from work that was done by Mark Carroll, who is another scientist in our lab, that they prefer the fresh pollen over the stored pollen that's been stored for a while. So basically immediately after they lay it down in the cell, they would prefer to have that and consume that compared to the pollen that's been stored for a while. However, that facet of, whether you change the source and it's, unbalanced, I don't know how that would factor in. I do know that, our recent experiments that I didn't talk about here, the foragers don't seem to discriminate. Okay, so they don't care. Like they do go after certain types of, let's say, diets in a very broad sense. But what they prefer is not dictated by omega fatty acid balance. It's actually more dictated by the levels of protein and lipid. So they like to go after things that have a lot of protein in them. So even though that might not necessarily be the thing that's most nutritious for them, the foragers will collect that. So that's a little bit, it's an interesting facet that you kind of brought up there.
Do they always store the same kind of pollen in a particular cell, or do they mix it in a cell?
They mix it. Yeah, right.
How does a mere beekeeper judge whether the pollen in a frame is good and healthy?
Okay, so one thing is if you see a lot of different colors, if that's a possibility, that's telling you that it's from a mixed floral source. But a lot of times, especially in Tucson, it's all one color because there's really only, we have a very limited floral environment, at many times of the year. So, a lot of times that's all one color. Just by looking at it, I don't think you can tell.
Can you tell by taste?
Oh, by taste. I don't know.
I find it's peppery when I taste it.
Oh.
I wonder if there's any chance that a human could tell whether pollen would be good, judging for the bee's needs.
I don't know. I think that, if you have...
Would it taste bad?
Yeah. I wonder if they, try to wall it off or something, if that's a signal that it's not very good. But, there's those stories of that entombed pollen, but that's more like,
Isn't that normally pesticide based?
Yeah, I think that's more of a pesticide thing. Yeah. But as far as nutrition,
how can we tell if pollen is entombed pollen? Is it just the cells that look black or what?
They put a cap on them.
Yeah, they put a cap on it.
I see.
Huh. Yeah, but that's more of like a pesticide thing. You're right. So I don't know. I don't know the answer to your question. Ask me in like 25 years.
Okay, we'll put you on the schedule for 25 years.
Okay. Yeah.
All right, I think that about exhausts the list of questions that I came up with.
Okay.
That was a great presentation, Vanessa. Thank you very much.
Okay, well, thank you for paying attention and for sticking with me for, over an hour now. So I appreciate, everyone coming and asking questions and the discussion. And
I really, really enjoyed it. Thank you so much.
oh, great. Thank you. And really, if
Likewise.
anybody, I'm happy to pass along those papers, Jerry, if you wanted to distribute them, talking about the
Please,
different.
that would be great.
Okay,
Thank you.
yeah, I can do that.
We can put links to on the website or make them available in our library.
Yeah, for sure. Because some of them are paywalled and I don't want you to have to
Yes.
worry about
please.
that. Yeah. And we should be having our paper, well, hopefully if the reviewers are okay with my my fixes that they suggested, you should have a publication talking about exactly what omega fatty acids are in 5 different supplemental diets.
That would be great. And I really appreciated that there is something that we can actually play around with, like adding flaxseed oil and that.
Yeah.
And
Yeah.
thank you for that. That was worth the price of admission.
Even though it's free.
Yeah. Right, we're sitting here for over an hour.
No,
So
it
time-
was a great presentation, and thanks
Thank
also
you.
thanks also
Great.
to Meg for the
Yeah.
work. We're looking forward to the results of your studies. They sound really interesting. So
Thank you
very
so much.
Yeah.
Yeah, Meg's a superstar. We're happy to have her. We're lucky.
Thank you both.
All righty.
All right.